Welcome to Bookfulness, your deep dive into the foundational texts of evolutionary science. This is episode 15. Last time we were really getting into the weeds with genes and genomes, that whole world of genetic information.
Right, the raw material. And today we're taking the next big step. We're moving from that blueprint, the genome sequence, to the actual organism. How do you get from A to B?
Okay.
And our mission today is to explore that connection using evolutionary developmental biology. Evo-Devo, for sure.
Evo-Devo, right. So it's about how the process of development itself changes over evolutionary time.
Exactly. How changes in development lead to, well, all the different shapes and forms we see out there. The morphology.
The whole reason this field even exists, it seems, comes down to something, well, kind of mind-blowing that Darwin himself picked up on.
Oh, yeah. He highlighted work by Carl Ernst von Baer, the big embryology guy back in the 19th century. Von Baer noticed something pretty astonishing.
Which was?
He looked at the embryos of different vertebrates. You know, lizards, birds, mammals. And in the really early stages, they were basically identical. You could hardly tell them apart.
Wow. Like a common starting point.
It's one of the most compelling pieces of evidence for common ancestry, honestly, outside of the DNA itself. It's visually right there.
And you see hints of that shared past in our own development, don't you?
Yeah.
Things that pop up and then disappear before birth.
Oh, absolutely. Human embryos develop these things called pharyngeal clefts. They look a bit like primitive gill structures.
Right.
And we briefly grow a tail, which then shrinks and fuses into the costylid.
We even get that fuzzy hair coat, lanugo.
Exactly, the lanugo, usually shed before birth. These aren't things we need as humans now, but they're leftover developmental steps from ancestors who did. They're sort of obligatory phases.
So Evo-Devo really starts with this shared beginning, this basic toolkit. And the big question is, how does that same set of initial instructions lead to a bird versus a fish versus a human? How do little genetic tweaks create wings or fins or hands?
That's the core puzzle. And the answer, as we'll dig into, isn't so much about inventing totally new genes all the time.
Okay.
It's more about changing how the existing genes are used, you know, when they turn on, how strongly, where they act, tweaking the controls.
Got it. And just a quick note for you listening as we go through some of these examples, we'll mention figures like figure 15.1 showing those similar embryos. You can find all those visuals in the supplementary file that comes with this deep dive. Just check the description. Okay, so let's start with those foundational patterns, the observations that really launched the field. Von Baer's law seems like the place to begin.
Definitely. Carl Ernst von Baer, writing way back in 1828, his key observation, his law, is that the features common to a large group, like all vertebrates, tend to appear earlier in development than the features that are specific to smaller groups, like just mammals or just a particular family or species.
So, okay, if I'm understanding this, it means every vertebrate embryo, whether it's destined to be a whale or a chicken, first builds the basic vertebrate stuff, like a backbone, a head, maybe some limb buds.
Exactly. That general body plan comes first.
And then it starts adding the whale-specific bits or the chicken-specific bits.
Precisely. The general blueprint precedes the specific details. It makes sense, right? Evolution tends to modify the later stages of development because tinkering with the very early fundamental steps is much riskier.
Right. You mess with the foundation, the whole thing collapses.
Pretty much. Changing the start of a complex process usually has disastrous effects downstream. So those early stages are highly conserved, very similar across broad groups.
This makes me think of that phrase you sometimes hear, ontogeny recapitulates phylogeny. Haeckel's idea.
Ah, yes, Ernst Haeckel. That idea sounds catchy, but it's largely an oversimplification and frankly misleading.
Why misleading?
Well, Haeckel implied that, say, a human embryo literally goes through stages where it looks like an adult fish, then an adult amphibian, and so on. That's just not true.
Okay.
A human embryo might have pharyngeal clefts that resemble structures in a fish embryo, but they never look like the fully functional gills of an adult fish.
And embryos develop their own unique features too, right?
Aye.
Things needed just for being an embryo that weren't in any adult ancestor.
Absolutely. The placenta in mammals is a perfect example. It's a complex adaptation purely for embryonic life inside the mother. It doesn't represent some adult stage from our past. Development isn't just replaying history, it's an active process shaped by selection for survival at that stage.
Okay, so development gets modified, not just replayed. And Evo-Devo points to three main ways these modifications happen, driving morphological change. You mentioned allometry, heterochrony, and heterotypy.
That's right. Let's tackle allometry first, allometric growth. This is about different parts of an organism growing at different rates during development.
Different rates, so it's not just getting bigger overall, it's about changing shape, because some parts grow faster or slower than others.
Exactly. It's about relative growth leading to shape change. The book gives that equation, why not exactly?
Yeah, the math bit. Let's unpack that.
Okay, think of X as maybe overall body size, like length, and Y is the size of some specific part, like the head or a limb.
Got it.
The key is the exponent, A. That tells you the relative growth rate of part Y compared to the whole body X.
So if A equals 1, they grow in perfect proportion, like scaling up a photo.
That's isometry, yes, perfect scaling. But if A is greater than 1, that's positive allometry. Part Y grows faster than the body. It gets disproportionately large.
And if A is less than 1, it's negative allometry.
Right.
The part grows slower than the body.
Precisely. It becomes relatively smaller as the organism grows.
We see this in humans, right?
Yeah.
After birth, our heads grow relatively slowly,
Negative allometry.
while our legs shoot up.
Positive allometry. That change in relative growth rates transforms our body proportions from baby to adult. It's all driven by these A values.
And small changes in A can make huge differences between species.
Huge differences. Look at the male stag beetle. Cyclomatus metallifer, 15.2 shows it well. The A value for their mandible growth relative to body size is way bigger than 1.
So a slightly bigger beetle gets enormously bigger jaws.
Exponentially bigger. It's extreme positive allometry. The largest males have these ridiculously oversized mandibles purely because of that accelerated relative growth rate. A small tweak in developmental rate creates this massive morphological weapon.
Wow. Okay, so that's changing the rate. What about changing the timing? That's heterochrony.
Yes, heterochrony is all about changes in the timing or rate of developmental events relative to each other or relative to the organism's overall lifespan, shifting the developmental schedule, basically.
And the classic example here has to be the axolotl, right? The Mexican salamander.
The axolotl is the poster child, absolutely. It shows a specific type called neoteny.
Neoteny, meaning it keeps juvenile features as an adult.
Exactly. It's a form of paedomorphosis retaining larval or juvenile traits into adulthood. The axolotl stays fully aquatic its whole life, keeps its external feathery gills, and never goes through metamorphosis like most related salamanders do to become land-dwelling.
So evolution didn't change the basic salamander body plan, it just stopped the clock before the final transformation.
Pretty much. It altered the timing of sexual maturation relative to body development. It matures while still looking like a larva.
Why? What's the mechanism?
It comes down to hormones. The axolotl fails to produce enough thyroxine.
Ah, the thyroid hormone that triggers metamorphosis in amphibians.
That's the one. All the genes needed to build the adult terrestrial form are still there, perfectly functional. The system just doesn't get the signal to switch over.
And we know the genes are functional because...
Because if you inject an adult axolotl with thyroxine, boom. It undergoes metamorphosis. It absorbs its gills, develops lungs, changes its skin, and turns into a typical terrestrial salamander. It proves the block is purely regulatory, a failure to produce the trigger hormone at the right time. Evolution just delayed that hormonal signal indefinitely. A timing change created a whole different life history.
That's incredible. The potential is locked away by a change in timing. Okay, third pattern, heterotypy. We've done rate, we've done time. This must be location.
You got it. Heterotypy is an evolutionary change in where a particular feature develops, taking a structure or process that normally happens in one place and shifting it to another.
The plant examples seem really clear, like that monstera vine growing roots out of its stem high up in the air.
Right, aerial roots, or the bryophyllum plant that grows whole new baby plantlets right on the edges of its leaves, roots and shoots appearing in totally unexpected places.
And in animals.
Think about things like osteoderms, bony plates developing within the skin of armadillos or crocodiles. Skin isn't the usual place for bone formation, so that represents a shift in where the bone-making developmental program gets turned on.
But the example everyone seems to love is the panda's thumb.
Ugh, the panda's thumb. It's a fantastic case of heterotypy and co-option working together. It's not a true finger, of course.
No, it's that modified wrist bone, right? The radial sesamoid.
Exactly. A small bone, normally embedded in the wrist tendons, has become dramatically elongated and repositioned to act like an opposable digit for gripping bamboo.
So the developmental program for bone growth and elongation got activated or sort of recruited to act on this specific wrist bone in a new way.
Precisely. The location of significant bone growth shifted, leading to this novel structure. It shows how evolution can repurpose existing parts by changing where developmental processes occur. Rate, timing, location, these are the fundamental ways development gets tweaked.
Okay, so we see these patterns changes in rate, timing, location. But how does evolution actually achieve these changes at the genetic level?
Hmm.
You mentioned earlier it's less about new genes and more about gene activity.
Right. Remember, almost every cell in your body has the same set of genes. The differences between a nerve cell and a muscle cell, or between a wing and a leg, come down to which genes are turned on or off, when, and how strongly. It's all about gene regulation.
And the big clues came from studying mutations that caused really dramatic body plan mix-ups.
Exactly, homeotic mutations. These are mutations where one body part is transformed into the likeness of another.
Like the classic fly example, turning an antenna into a leg or something.
That's a famous one, yes. But the one that really opened the floodgates was the ultrabithorax, or UBX, mutation in fruit flies Drosophila.
Okay, UBX. Normal flies have two wings on their middle segment, the T2 segment.
Right.
And then tiny little balancing organs, halters, on the next segment back, T3.
Correct. The UBX homeotic mutation messes that up. It transforms the T3 segment, the halter segment, into another copy of the T2 segment, the wing segment.
So you get a four-winged fly.
Exactly. A fly with a second pair of fully formed wings where the halters should be. Figure 15.8 shows it. It's quite startling.
And what did that specific mutation tell scientists?
It was a revelation. It showed that the UBX gene wasn't just coding for, like, one protein needed for the halter. It must be a master control gene.
The master switch.
Yes, a gene whose job is to control a whole suite of other downstream genes, hundreds of them, probably that collectively determine the identity of the entire T3 segment. Turning UBIC off in T3 flips the switch from make a halter segment to make a wing segment.
Whoa, okay. And this led to the discovery of a whole family of these master genes.
It did. The Hox genes. These are the key homeotic selector genes. They encode transcription factors, proteins that bind to DNA and regulate other genes.
And their main job is?
Their fundamental role, conserved across the animal kingdom, is to specify identity along the anterior-posterior axis, the head-to-tail axis. They tell developing segments you are head segment 1, you are thoracic segment 2, you are abdominal segment 5, and so on.
Okay, so they lay out the basic body map, found in flies,
Oh.
but you say conserved.
That was the truly mind-blowing discovery. Hox genes aren't just in flies, they're in practically all animals. Everything from jellyfish and worms to fish, frogs, mice, and us, except maybe sponges, which lack that clear body axis.
So these same master control genes evolved way back.
Over 540 million years ago in the common ancestor of most animals alive today. They set up the fundamental body plan, and that system has been maintained ever since.
500 million years. That's incredible conservation.
It really is. And generally, the genes are lined up on the chromosome in the same order that they are expressed along the body axis from head to tail. It's called collinearity.
And mammals, we have them too.
We do. We actually have 4 sets due to some ancient whole genome duplications in our vertebrate lineage.
Yeah.
But they still line up, and they still specify regional identity along our spine during embryonic development. Neck region, chest region, lower back region. It's the Hox code at work.
Okay, so this brings us to the idea of the genetic toolkit. This shared set of fundamental developmental genes. Hox genes being prime examples that pretty much all animals inherit from that ancient ancestor.
Exactly. A common toolbox used to build an incredible diversity of forms.
But if everyone's using roughly the same tools, how do you build such different things? How do you get novelty? You mentioned co-option earlier.
Co-option is key. It means taking an existing gene from the toolkit, one that already has a job, and recruiting it or co-opting it for a brand new function in a different time or place during development.
Ah, so you don't need to invent a whole new gene for, say, making fingers.
Right. You might take a gene that was originally involved in setting up body segments and find a way to turn it on in the developing limb bud to help pattern the fingers.
It's like repurposing a tool, using a screwdriver as a chisel sometimes.
Sort of, yeah. It's evolutionary tinkering at its finest. Why reinvent the wheel when you can just use an existing part in a novel way? It's often achieved by changing the gene's regulation, not the gene's protein product itself.
Why is changing the regulation safer?
Because these toolkit genes, especially transcription factors like Hox genes, are often pleiotropic.
Yeah.
They have many different jobs in many different parts of the body at different times.
Okay.
If you mutate the protein itself, you might mess up all its jobs, which could be lethal. But if you just change a specific regulatory switch and enhancer that turns it on in a new place, you can potentially gain a new function without disrupting the old ones.
That makes sense. Modularity.
Yeah.
Can we see this co-option with limb evolution?
Limb evolution is a prime example. In ancestral fish, certain Hox genes specifically, a group from the Hoxa and Hox clusters, like genes 9 through 13, were mainly involved in patterning the main body axis.
Okay.
But with the evolution of tetrapods, four-limbed vertebrates, and the origin of limbs, especially the autopod, that's the hand-foot part, with wrists, ankles, and digits,
Yeah.
these same Hox genes were co-opted. They got turned on in the developing limb bud, out away from the body axis.
So they started patterning the limb from shoulder to fingertip.
Exactly. They were recruited to specify the different segments along the proximal distal axis of the limb. And this likely happened because new enhancer sequences evolved near these Hox genes regulatory switches that responded to signals within the limb bud, turning the Hox genes on in that new location.
So the evolution of hands and feet wasn't about inventing hand genes. It was about redeploying the existing body segment genes in a new context.
That's a great way to put it. Novelty often arises from new combinations and new regulations of ancient toolkit components.
Okay, so that co-option idea, using old genes in new ways, leads us right into thinking about how gene regulation evolves. You're saying a lot of morphological evolution isn't about changing the proteins the genes make, but changing the switches that control them.
That's the central idea, often called the regulatory hypothesis of morphological evolution. Much of the diversity we see seems to stem from changes in cis-regulatory elements, or CREs.
Cis-regulatory,
Yeah.
meaning they are on the same DNA molecule near the gene they control.
Exactly. These are stretches of DNA, often called enhancers or silencers, that act as binding sites for transcription factors. They function like on-off switches, or maybe dimmer switches, controlling when, where, and how strongly a gene is turned on, transcribed.
And these enhancers can be quite far away from the actual gene sometimes, right? Not right next door.
Oh yeah, they can be thousands, even millions of base pairs away, looping around in 3D space to interact with the gene's promoter region. The key is they are physically linked on the same chromosome, hence cis.
So changing one of these enhancers, one specific switch, is less risky than changing the gene's main code.
Much less risky, generally, because an enhancer often controls gene expression in only one specific tissue or time point. So mutation in that enhancer might affect the gene's function just in the developing wing, for example, leaving its function in the eye or the gut completely untouched.
Ah, that modularity again. It allows for fine-tuning in one place without breaking everything else.
Precisely. It avoids the problem of pleiotropy, one gene having many effects.
Can we see this with trait loss, where losing an enhancer causes a feature to disappear?
The classic example is the three-spined stickleback fish, Gasterosteus. We mentioned them before.
Right, the ones that lose their pelvic spines in freshwater.
Exactly. Marine sticklebacks have these prominent bony spines projecting from their pelvis, basically remnants of hind limbs used for defense. But many freshwater populations have lost these spines, sometimes very rapidly after colonizing a new lake.
And how did they lose them?
Research pinned it down to mutations in a specific enhancer region for a gene called PIC1.
PIX1, what does that gene normally do?
PIX1 is a crucial transcription factor involved in lots of developmental processes, including hind limb development, but also jaw formation, pituitary gland function, really important stuff.
Okay, so mutating the PIX1 protein itself would be bad news.
Very bad news, likely lethal. But what happened in these freshwater sticklebacks is that they lost the function of one specific enhancer that is solely responsible for turning PIX1 on in the pelvic fin primordium, the developing spine bud.
So the switch for the pelvis got broken.
Exactly. PIX1 expression disappears specifically in the pelvis, so the spines don't develop. But because only that one enhancer is affected, PIX1 continues to function perfectly normally in the jaw, the pituitary, everywhere else it's needed.
That's incredibly neat. Targeted trait loss through enhancer deletion? Figure 15.15 illustrates this, right?
It does. It's a beautiful demonstration of how evolution can use cis-regulatory mutations to fine-tune adaptation locally without causing harmful side effects elsewhere. Decoupling allows precision.
Okay, that's loss. What about gaining new traits through changes in enhancers.
We see that too. A great example comes from wing pigmentation patterns in Drosophila. Some species have evolved novel, dark spots on their wings, often used in courtship displays.
So a new spot appears where there wasn't one before. How does an enhancer drive that?
In several cases, researchers found that these new spots evolved because of the emergence of a new enhancer sequence near a pigmentation gene, or sometimes near a gene like wingless, which influences downstream patterning.
A new enhancer. How does that just appear?
Well, DNA sequences are always mutating. A series of point mutations, insertions, or deletions in a non-coding region could, by chance, create a sequence that happens to be recognized and bound by an existing transcription factor that is active in that part of the wing.
Ah, so an existing transcription factor finds a new binding site that wasn't there before.
Exactly. And if that binding activates a gene involved in making pigment, suddenly you get pigment deposited in a new spot on the wing. Figure 15.16 shows an example related to the yellow gene.
So evolution essentially created a new regulatory address for an existing gene, leading to a novel pattern.
Precisely. Again, it's modifying the control system, not the core machinery, to generate evolutionary novelty. Cis-regulatory evolution is a major engine of morphological change.
Okay, so we've covered changes to the switches, the cis-regulatory elements, but what about changes to the things that flip the switches, the transcription factor proteins themselves?
Right, that's the other side of the regulatory coin. Transregulatory changes. These are mutations that alter the transcription factor proteins. Transverts because these proteins can diffuse through the cell nucleus and act on genes located on different chromosomes, or far away on the same one.
So instead of changing the lock, the enhancer, you're changing the key, the transcription factor.
That's a good analogy, yes. You modify the protein itself. Maybe you change which DNA sequences it can bind to, or how strongly it binds, or which other proteins it interacts with to activate or repress gene expression.
This sounds potentially more consequential than changing just one enhancer.
It often is, because that one transcription factor might regulate dozens, hundreds, or even thousands of target genes throughout the genome. If you change the protein, you potentially alter its interaction with all of its targets.
Higher risk of unintended consequences then. Pleiotropy again.
Exactly. That's why many researchers think cis-regulatory changes might be a more common path for subtle morphological tweaks. But transregulatory changes, while potentially riskier, can sometimes lead to really major fundamental shifts in the body plan.
Like the origin of the insect body plan. Going from many-legged ancestors to just 6 legs on the thorax.
That's a fantastic example. Ancestral arthropods, like modern crustaceans or myriapods, typically have legs on most of their body segments, including the abdomen. Insects are defined by having legs restricted to the 3 thoracic segments.
So how did they lose the abdominal legs?
A key player, again, is the Hox gene altered the thorax, UBX. In insects, UBX is expressed in the abdomen, starting in the first abdominal segment.
Okay.
And crucially, the insect version of the UBX protein evolved a new capability compared to its ancestors, like in crustaceans. It gained the ability to actively repress the genes required for leg development.
So where UBX is turned on in the insect abdomen, it actively shuts down the leg-building program.
Precisely. It acts as a repressor of legs in that region. And comparative studies show that the UBX proteins in crustaceans and other related groups lack this strong leg-repressing activity.
What changed in the protein to give it this new function?
Research points to the evolution of specific new motifs or domains within the UBX protein sequence in the insect lineage. One often cited is a polyalanine-rich region near the C-terminus, which seems to contribute to its repressive function, possibly by altering how it interacts with other co-repressor proteins. Figure 15.18 touches on this.
Wow. So a modification of the UBX protein itself, a transregulatory change, was fundamental in defining the entire insect body plan. 6 legs good, abdominal legs bad.
It appears to be a critical piece of that puzzle, yes. A change in the key led to a massive morphological reorganization.
Are there other examples where changing the transcription factor protein leads to a radical shift?
Yes, work on another Drosophila segmentation gene, Fushitarazu, provides some really compelling experimental evidence.
Fushitarazu, Japanese for not enough segments, right?
Yeah.
Because mutations cause segment loss.
That's the one. It plays a role in establishing the repeating segment pattern early in development. Researchers noticed that the NTC protein in Drosophila was missing a particular short amino acid sequence, a motif called the YPWM motif.
Okay.
This motif is involved in protein interactions, and it is present in the SE's protein of more ancestral insects like the flower beetle Tribolium.
So the Drosophila version lost this interaction capability.
It seems so. And they did experiments to see what effect that had. First, they genetically engineered Drosophila st to put that YPWM motif back in.
And what happened?
Even just adding back that short motif caused a partial homeotic transformation. Parts of the fly's antenna started developing characteristics of a leg.
Just from adding that little interaction sequence.
Yeah, but then they did the full swap. They replaced the entire Drosophila ft's gene with the st's gene from the flower beetle. The result was a complete homeotic transformation of the antenna into a leg. The fly grew a recognizable leg, complete with the typical 5-segmented tarsus at the end, right where its antenna should have been. Figure 15.5 line shows this.
That's incredible. It totally switched the identity of that body part just by swapping one version of a transcription factor for another ancestral version.
It powerfully demonstrates how changes within the transcription factor protein itself, altering its interaction domains and therefore its regulatory function, can lead to really dramatic wholesale changes in developmental outcomes. Transregulatory evolution can be a potent force for major morphological shifts.
We've talked a lot about how evolution can generate novelty by tweaking rates, timing, location, cis-regulation, transregulation.
Yeah.
But it feels like we also need to talk about the limits. Evolution isn't all-powerful, right? There are constraints.
Absolutely. This brings us to concepts like evolvability and developmental constraint. Evolvability is, sort of, the capacity of a trait or a lineage to generate adaptive variation and evolve in response to selection.
It depends on having genetic variation, obviously.
Yes, additive genetic variance is crucial. But evolvability also depends heavily on how different traits are genetically connected or correlated. This is where phenotypic integration comes in.
Phenotypic integration, that's the idea that traits that function together tend to be genetically linked.
Often, yes. Think about the different parts of a bird's beak, the upper mandible, maxilla, and the lower mandible. They have to work together seamlessly to grasp food.
Right.
Very often, the genes that influence the length of the maxilla also influence the length of the mandible. This might be due to pleiotropy, one gene affecting both parts, or close genetic linkage. So their development is integrated.
And this integration can be helpful sometimes, but restrictive other times.
Exactly. It has a dual nature. If selection favors a longer beak overall, having a positive genetic correlation where both parts tend to get longer together facilitates that evolutionary response. Selection pushes on one part and the other comes along for the ride easily.
Makes sense. Faster adaptation.
But what if selection favors an unusual beak shape? Like a very long lower mandible, but a short upper mandible.
That genetic correlation now works against it, right? It makes it hard to change one part without changing the other in the same direction.
Precisely. The integration now acts as a constraint, slowing down or potentially preventing evolution towards that specific shape. To achieve such an unintegrated shape requires mutations that specifically break or reduce that genetic correlation.
And you mentioned the black skimmer bird earlier with its longer lower jaw. That must have been a case of overcoming such a constraint.
It's a fantastic example of breaking the mold. It achieved a morphology that goes against the typical integrated pattern seen in almost all other birds. It shows constraints can be overcome, but it might take specific genetic changes or strong, persistent selection.
So this leads us to developmental constraints more broadly. These aren't just genetic correlations, but fundamental biases in the developmental process itself.
Right. Developmental constraint refers to biases in the production of phenotypic variation. Development isn't capable of producing all conceivable forms with equal ease. Some variations arise readily, others rarely or never.
It explains why certain imaginable creatures just don't exist. Like the book mentions, we never see a tetrapod, a four-limbed vertebrate, where the middle digit, our middle finger, is the shortest one on the hand or foot.
Yeah, that specific pattern seems to be developmentally forbidden or at least extremely difficult to produce across all tetrapods. The way digits form imposes a bias against that particular arrangement.
There was a classic experiment demonstrating this kind of inherent bias, wasn't there, with frog and salamander limbs?
Yes, the Albert and Gale experiment. It's a cornerstone study. They took developing limb buds from frogs, Xenopus, and salamanders, Ambystoma, and they treated them with a chemical called colchicine, which inhibits cell division, mitosis. This basically stunted the growth of the limb bud, forcing it to develop with fewer digits than normal.
So they artificially created limb reduction.
Yeah.
And what did they find?
They found remarkably consistent but different patterns of digit loss in the two groups. In the frogs, the treatment consistently led to the loss of digits from the front edge of the limb, the preaxial side, like losing the thumb first.
Okay.
But in the salamanders, the treatment consistently caused digit loss, starting from the back edge, the postaxial side, like losing the pinky first.
Whoa, so the same disruption caused opposite patterns of loss. Why is that significant?
Because those experimentally induced patterns perfectly matched the patterns seen in natural evolution. When frogs evolve reduced digits naturally, they tend to lose the preaxial ones. When salamanders reduce digits, they tend to lose the postaxial ones.
Ah, so the experiment revealed an underlying developmental bias. The way their limbs are built makes certain patterns of reduction much more likely than others?
Exactly. It showed that the path of least resistance for developmental change was different in the two groups, constraining the direction of their evolutionary limb reduction. It's a direct demonstration of developmental constraint shown clearly in figure 15.25.
That's powerful. But perhaps the most striking example of a long-term developmental constraint is the number of neck bones in mammals.
Oh, the 7 cervical vertebrae is just astonishing. Almost every single mammal, from a tiny shrew or bat to a blue whale, and yes, even the giraffe with its incredibly long neck.
7. They all have just 7 neck vertebrae. Why? Surely a giraffe could use more.
You'd think so. It seems wildly counterintuitive. The explanation appears to be extreme developmental constraint linked to severe pleiotropic effects.
Meaning changing that number causes other problems.
Catastrophic problems, it seems. Studies of human developmental abnormalities show that attempts to change the number of cervical vertebrae, like having the 7th vertebra develop ribs like a thoracic vertebra, or the first thoracic vertebra lacking ribs like a cervical one, are very strongly associated with a higher risk of pediatric cancers and other severe congenital malformations.
Like what kind of malformations?
Problems with the heart, lungs, skull, nervous system development. The genes controlling the identity of those cervical vertebrae, the Hox genes specifying that region,
The Hox genes again.
they seem to be deeply intertwined with the patterning and development of many other critical organ systems early on. Messing with the cervical count seems to disrupt this larger network in ways that are almost always highly detrimental, often lethal.
So natural selection has just ruthlessly eliminated any variation in that number for hundreds of millions of years because the side effects are too severe.
That appears to be the case. It's not physically impossible to build a mammal with 6 or 8 neck bones, but the developmental system is set up such that doing so almost invariably breaks other essential things. The developmental cost is prohibitive. It's a profound constraint.
Okay, so constraints limit evolution. But what about the opposite? When development is flexible, that's phenotypic plasticity, right?
Exactly. Phenotypic plasticity is the ability of a single genotype, one set of genes, to produce different phenotypes, different observable traits in response to different environmental conditions.
One genotype, multiple potential outcomes depending on the environment.
Precisely. We often visualize this with a reaction norm. You plot the environmental variable, like temperature, on the x-axis, and the resulting phenotype, like body size, on the y-axis.
So a genotype that's not plastic would just be a flat horizontal line. Same phenotype regardless of environment.
Right, that would indicate strong canalization, which is sort of the flip side of plasticity. Canalization is the evolution of developmental pathways that are buffered against environmental and sometimes genetic variation, producing a consistent, stable phenotype. Robustness.
Okay, so canalization resists change, plasticity allows change.
And a highly plastic genotype would have a steep slope on that reaction norm graph. The phenotype changes dramatically with the environment. Think about those aquatic plants again.
Yeah, the ones with different leaf shapes depending on whether they're underwater, at the surface, or in the air.
That's extreme plasticity. The same genes produce totally different leaf morphologies triggered by signals like light levels, water contact, or hormones, allowing the plant to optimize function in each microhabitat.
So plasticity can be adaptive, allowing organisms to cope with variable conditions.
Definitely. But it also led to a really fascinating idea pushed by Conrad Waddington back in the mid-20th century: genetic assimilation.
Genetic assimilation. Does that mean a plastic trait becomes, like, hardwired genetically?
That's the core idea. Waddington proposed and demonstrated that a phenotype initially produced only in response to a specific environmental trigger could, through selection, eventually become the default phenotype, expressed even without the original trigger.
How did he show that? The famous fly experiment.
The famous heat shock experiment with Drosophila. He took fly pupae and gave them a heat shock. This environmental stress sometimes disrupted normal wing development, causing a specific defect: the absence of a small crossvein. A cross-veinless phenotype.
Okay, so heat caused a weird wing. That's plasticity.
Right. Initially, only heat-shocked flies showed it. But then Waddington did something clever. He selectively bred only those flies that showed the cross-veinless phenotype after being heat-shocked. He picked the most sensitive ones each generation.
So he selected for the ability to respond to heat shock in that specific way.
Exactly. He kept doing this for many generations. And the crucial result was that eventually some flies started developing the cross-veinless wing even when they were raised at normal temperatures without any heat shock at all.
Whoa, the trait became genetic. It didn't need the environmental cue anymore.
Precisely. It had been genetically assimilated. Waddington's interpretation was that the initial heat shock revealed cryptic genetic variation present in the population, combinations of genes that predisposed flies to making cross-veinless wings, but which were normally hidden or buffered by canalized development.
The heat broke the buffering.
It overwhelmed the buffering, allowing selection to see and favor those underlying gene combinations. Eventually, selection accumulated enough of those genes that the trait became expressed constitutively without the need for the environmental stressor.
That's a wild idea. It suggests the environment can sort of guide evolution by first inducing a trait plastically?
It does, and this connects to modern debates about the role of plasticity in adaptation, particularly Mary Jane West Eberhard's hypothesis that plasticity might often precede genetic adaptation.
The plasticity first or paving the way idea.
Yeah. The idea is an organism enters a new environment. Its existing plasticity allows it to immediately produce a somewhat adaptive phenotype, maybe not perfect, but good enough to survive. This buys time.
Buys time for what?
Buys time for selection to then favor mutations that refine, stabilize, or genetically accommodate that initially plastic phenotype, making it more efficient or reliable. So plasticity initiates the adaptive response, and genetic evolution follows up to solidify it.
That sounds plausible, but is that always what happens? Or does genetic change sometimes just compensate?
That's the critical question. Does genetic evolution build upon the plastic response to create something new, accommodation, or does it simply act to counteract the environmental effect to maintain the original optimal phenotype, compensation?
Compensation would mean plasticity isn't really leading the way to novelty.
Right. It's more like genetic evolution is just restoring the status quo against environmental pressure. And there's good evidence for compensation, too.
Like the frog tadpole example.
Exactly. The study comparing green frog tadpoles, rhoniclamitins, from cold high-elevation ponds versus warmer low-elevation ponds.
Cold water slows development down, so the high-elevation tadpoles needed to cope.
Right. And they did evolve genetically based, faster growth rates compared to the lowland frogs when measured at cold temperatures. This faster intrinsic rate helped them compensate for the cold, allowing them to reach metamorphosis before winter. It was adaptive.
Okay, so genetic evolution happened, but was it accommodation or compensation?
Well, when they raised tadpoles from both populations across a range of temperatures, including optimal warm temperatures, both populations actually showed the same maximum growth rate at that optimum.
Ah, so the high-elevation frogs didn't evolve to be super growers overall. They just evolved to grow faster, specifically in the cold, to match the performance level the lowland frogs achieved easily in the warmth.
Exactly. The genetic change compensated for the environmental handicap, cold, restoring fitness back to the species' typical maximum, rather than building upon plasticity to achieve some new, higher level of performance. The reaction norms shifted, as shown in figure 15.29, but the peak performance remained the same.
So in this case, at least, genetic evolution seemed to be playing catch-up to the environment, not necessarily using plasticity as a launch pad for brand new adaptations.
It suggests compensation can be a major role for genetic change following environmental shifts, which means the general importance of the plasticity-first mechanism in driving major evolutionary innovation is still an active area of research and debate. It probably happens sometimes, but maybe not always. #outro. So wrapping up this deep dive into Evo-Devo, I think the big picture is just fascinating. We see this incredible diversity of life forms all around us.
Yeah.
And yet it seems to be built by tinkering, often in subtle ways, with a remarkably conserved, ancient set of genetic tools, like the Hox genes.
It's not always about inventing new parts, but finding new ways to use the old parts. And a lot of that comes down to changing the controls, the gene regulation.
Exactly. Changes in those cis regulatory enhancers allow for really Fine-grained, modular evolution changing one little bit without breaking the rest.
Like the stickleback losing its spines just in the pelvis.
Right. And then changes in the transregulators, the transcription factor proteins themselves, while maybe riskier, can lead to profound shifts, like fundamentally altering the insect body plan via changes in UBX.
But evolution isn't free to do anything. It's channeled by these deep developmental constraints.
Definitely. Things like the near-universal 7-mech vertebrae in mammals show how developmental history can lock things in place for eons.
And it's all playing out against this backdrop of flexibility versus stability, the interplay between phenotypic plasticity, the ability to respond to the environment, and canalization, the buffering that keeps development on track.
That tension shapes how organisms adapt and evolve.
It really makes you think, doesn't it?
Yeah.
When you consider those deep constraints like the neck vertebrae and the fact that so much difference comes from tweaking the timing and location of the same basic genetic toolkit. How much of the difference between you sitting here and, say, a chimpanzee or even a mouse is really just subtle shifts in developmental timing heterochrony applied to that shared ancestral instruction manual?
It's a profound thought. Beneath the vast diversity, there's this deep unity rooted in shared developmental pathways, stretched and squeezed by evolutionary time.
A really powerful takeaway.
Okay.
That wraps up Unit 3 of our journey through this text.
Indeed. And next time, we embark on our final major section, Unit 4, which is all about macroevolution and the history of life.
So resuming out, from development to the grand scale of life's history.
Exactly. We'll start by looking at how we reconstruct that history, how we build and interpret the tree of life itself, phylogenetics.
Sounds great. Well, thanks for joining us for this deep dive into the fascinating world of Evo-Devo.
We hope you'll join us next time as we start exploring the tree of life.